Exploring the effects of acoustic levitation on plant growth and development. Acoustic levitation is a technique that uses sound waves to suspend small objects, including droplets of liquid, in midair. While acoustic levitation has been used in various scientific fields, such as material science and physics, its effects on biological systems, particularly on plant growth and development, have not been thoroughly studied. The idea behind this experiment is to investigate how the absence of a solid support structure and exposure to continuous sound waves in a levitated state might influence the growth, morphology, and overall health of a plant. Plants grown in an acoustically levitated environment will exhibit altered growth patterns, including differences in root development, stem elongation, and leaf formation, compared to plants grown under normal gravity conditions with traditional soil or hydroponic supports. Acoustic levitation device capable of levitating small objects for extended periods. Seeds of a fast-growing plant species, for example, Arabidopsis thaliana or radish. Growth medium aerogel or nutrient gel that can be levitated along with the seed. Controlled environment chamber to maintain consistent temperature, humidity, and light. Standard plant growth setups for control experiments. Cameras and sensors for monitoring plant growth in real time. Analytical tools for assessing plant physiology, for example, chlorophyll content, root architecture analysis. Set up the acoustic levitation device inside a controlled environment chamber. Place the seeds in a small amount of levitatable growth medium aerogel or nutrient gel that can be suspended in the levitator. Set up control groups with seeds planted in soil or hydroponic systems under identical environmental conditions. Activate the acoustic levitation device and ensure that the seeds with the growth medium are levitated in a stable manner. Monitor the seeds daily, recording any changes in their position, orientation, and growth patterns. Measure the growth rate, stem length, root development, and leaf formation over time, comparing the levitated plants to the control groups. After a specified growth period, analyze the plants for differences in physical characteristics such as root architecture, stem thickness, leaf size, and overall biomass. Conduct physiological assessments such as chlorophyll content analysis and photosynthetic efficiency measurements to determine if the continuous exposure to sound waves and lack of physical support affects the plant's metabolic processes. Compare the results between the levitated plants and the controls to identify any significant differences in growth and development. Determine whether acoustic levitation has a measurable impact on plant growth and development. Assess the potential applications of this technique in space agriculture, where microgravity environments may benefit from alternative plant growth methods. Publish the findings, noting any limitations and suggesting further research avenues, such as exploring different plant species or modifying the acoustic levitation parameters. This experiment could provide valuable insights into the effects of non-traditional growth environments on plants, potentially opening up new avenues for agricultural practices in space or other challenging environments. The novelty of combining acoustic levitation with plant growth studies would likely make this a pioneering experiment in the field of plant science and biophysics.